Lot of various things you can do. There are so many softwares available in ExPASy. One of the important tool that is being used for understanding protein-protein interaction is the string database. The full form of string database is a search tool for the retrieval of interacting gene or protein. It's a database which is which is having the known and predicted protein-protein interactions. The interaction includes direct physical as well as indirect, which is functional association derived from various sources. For example, computational prediction, knowledge transfer between organisms, as well as interactions aggregated from various databases. So string is really important to understand if a protein is interacting with the other protein, and you can also understand the complex molecular signaling pathways using string. So in this one, what we will do is we will explore the website. We will add query proteins. We will see some of the basic network, and we'll also see what are the settings or analysis options that are available. Finally, we'll also see how we can export the image and create a nice, decent network of the protein with as much as accuracy we can have, right? So as you can see in string, it covers over 5,000 different organisms with over 25 million high confidence links between proteins, which is really, really important if you're working in the area of protein-protein interactions. So without any delay, let's move on to the resource website. Okay, so what I've done is you can type in ExPASy string in Google, and it will direct you to this particular link, which is string SIB, Swiss Institute of Bioinformatics. When you click on this one, you will be directed to this page, which is the page of the ExPASy, where you will have a tab which can direct you to the resource website of the string database. So once you click on this one, you should be able to open this, the main page of the string website, and you can see this is the logo of the string and some of the options that are available on the left side and some of the options that are available on the top side, and then this is the interface. Let's see what we can do. We can also do the login. We will register to the website. It's important. When you register to the website, the information goes into the string database, and it's important for the developers to know how many people are using, and it's going to help them to further advance or add multiple features. And also secure the funding for the database. So you can see here, there are a couple of options on the left side. We can start with protein by name. This is the first option, most basic option, and probably in most of the cases, you can directly use this option and get your results out. And second one, multiple proteins. So you can also add multiple proteins. As you can see over here, examples are also given. So you can type in the name of the proteins, and also you can also type in the UniProt ID of the protein. You can upload the file also, and you can mention the organism name right over here. There are some advanced setting options also. If you want to change the confidence level of the interaction, you can also change that. You can make it like highest confidence, which is 0.9, high confidence 0.7, and then medium confidence 0.4. So the low confidence 0.1, right? So probably you should not be going below 0.4, at least not below the medium confidence level. And then you have other options over here. Next, what you can do is protein by sequence. You can also add amino acid sequence, or you can also add the file of the protein's amino acid sequence. And then it can auto detect the protein, and you can also give the name of the organisms right over here. Right, protein with values, another option, protein families, and then you have multiple options over here also. And then pathway and diseases, this is important, and it's also different. You can type in a particular pathway. So for example, I'll take the example of, which are given over here. You have carbon fixation, example number 3, based on PubMed ID. Okay, so you can also give the ID of the research article. In this case, 4 is, cell cycle arrest. If I want to find out the proteins or the network of the proteins in this particular pathway, which is cell cycle arrest of Homo sapiens humans. I can directly click on the search tab and I'll get. As you can see over here, you can also add gene ontology term, get pathway. So similar like this, you can also add published manuscripts, and then get the network out. This is new feature. Add organism is also a new feature. Add any organism to string annotate proteome. So this is another feature. You can upload the proteome. Next is organism option is there. Directly by typing in the organism, you can do the search. Examples are also given over here. So if you want to see the examples, you can also explore some of the examples. So this is one example which is showing you how the network will look like. We'll go into the detail by our own query protein. So I'll go back. I'll not show this. Right. So I'll go back and then we'll put a particular protein of my interest. So I'm working with LASR protein. This is the protein which is essential in Pseudomonas aeruginosa quorum sensing signaling pathway. It's a regulator protein. It's a transcriptional factor which regulates the quorum sensing signaling pathway, and it also controls the expression of various genes. So that means it's associated with other proteins also. In this case, you can directly click on search, and it will auto detect all the organisms that are associated with that particular protein. So you can see over here, LASAR is written, and then organisms are also written over here. And this gives me a good idea that how many organisms might have this particular protein. That means that signaling is common with all these organisms. So that means LASR signaling is present in Pseudomonas aeruginosa. LASR signaling, similar signal is also present in other organisms, like it's also there in, which is closely related, agrobacterium also. Yeah. So you can see, you can find out the other organisms that are associated with a similar pathway, and then you select the organism that you're looking for, which is Pseudomonas aeruginosa. As you can see over here, it's been studied in more detail. So I'll press on continue. So it will detect the protein, it will detect the organism, and then you can actually see the network right over here. In the network, what you can see, this red node, it is the query protein, in my case LASR. So that will be red, and other proteins will have different colors. They will also have a crystal structure in the center. As you can see in all of them, we have a crystal structure in the center. That means the crystal structure is available in the database. It could be PDB database, it could be any other database where you can find the crystal structure. If you want to know more about the protein or the partner protein or the nodes, for example, as you can see over here, RHLI is another signaling system that is present in Pseudomonas aeruginosa. This is the receptor and this is the inducer. So they do have interaction. That means this receptor is also linked with the inducer protein. So as you can see over here, this is the LASR protein and associated with various other proteins also. Another signaling system is a PQS signaling system. It's also linked with PQS. LAS A is also there, another protein. VFR is also there. So many proteins are associated. And now we can go down and go to the setting where we can change few of the parameters. Now, this is the full network. You can also stick to just to the physical interaction also. Now, these are the proteins that are physically interacting. So you can see the number significantly decreased in this case. And if you want to see the full map, you can again update it and you can see the entire map again. Now, some of the proteins that are shown with evidence, and you can also change them into the confidence level. So you can see now basically the thickness of this line represents how much the confidence is there in the protein-protein interaction. So this is also a good way to represent the network. And then next, what we can see is you have other options like text mining, experiments, databases. If you want to just focus on experiments, let's see how many proteins we'll get just based on the experiments. Only one protein, right? So which is very, very unique. So you have to rely on various other parameters like neighborhood, gene fusion, co-expression, and then based on your preference, you can change this network. So it's very, very important how you customize this network and what information you want. Now, medium confidence level is that right over here as the default setting. You can increase this and update it. It's also going to change because now I have a lot of other parameters that I've included in here. Okay, let's go next. Now we want to see more than 50 interactions. Default is top 10. Now we can see more than 50 interactions. It's not updating because then interaction, we have to decrease the confidence level to medium. Then we are definitely going to see a lot of complex network in case of this particular signaling pathway. So you can clearly see how complex it became suddenly when I changed the partners and also lower down the confidence. So this is how you can basically create network of more than 50 interactions. You can also create shells, right? More than 50 interactions. Let's update this and see what happens. So depending upon how you want to, how much you want to understand, you can make this network really, really complicated. Confidence level is very important key. So you need to understand that. You can't just be adding, a lot of things in there and make this network too complicated, right? So you need to be careful on that side. So what I'm doing is I'm going to restrict this to 10 interaction because otherwise it's too difficult to handle all the proteins together. So I think this is good. So now we can move on further where we need to do some more analysis on this one. Rather, I'll make this network also a little bit more smaller because we need to see carefully that what is happening in our network. So now you can see in this network, we have lots of proteins from various signaling pathways. Now, if we go to analysis, we can categorize these into a different groups. We can color them. So you can put quorum sensing protein in red. So you can see these are all quorum sensing associated proteins. So many proteins are there, right? And then quorum sensing regulation is also linked with secondary metabolite biosynthesis processes. And we can link it with the modulation of, let's see, extracellular region, which is green. And then you can see that now the network is becoming more and more useful for us, and you can randomly just change this, right? So based on this, you can show your network in more interactive way, where just seeing at the network, you'll get a lot of information out. So you can customize this network according to your needs and the options that are available in the setting, right? So you can see over here, this particular quorum sensing protein is also showing red color, and also the blue color is or the violet color is there, same in this case. So they have a dual function in this case. Now next, the same thing you can do if you want to find out virulence and virulence associated, which is, I think, one of the important parameters. So you can see all those proteins that are virulence factors that are actually causing the damage to the host. So this is important. Now, once you're happy with all these settings, you can go to export, and in export, you will have option to export the image in various formats. And what I want right now is just the format in the PNG format of the protein, which is the image format. And I'll click, and now you can see this entire network where all the proteins are highlighted. The background is not there, so you can directly paste it in any file, and as you can see, the diagram the background is not there. So because of that, you should be able to see all the interactions very nicely. Here the background is dark. It will look like this on a white background, so which is good, right? So you can export other formats also, and then use those images in your basically in your publications and in your reports also. Now there are some other resources that I want you to explore, which is here in the help center. In help center, if you go to string videos, these are very nicely formulated videos, and you will get a lot of information from these videos. What you have to do is just watch these videos and follow along. Basic tutorial is given, and then if you want to go and do the network visualization, improve visualization, everything is available. Frequently asked questions are also available. What are the scores and how it works, which is nice, which is good to understand. And I think once you have the data out, you should be able to understand the data. You should be able to report the data in your publications, and hopefully you will be able to get your papers accepted by good journals. But obviously, when it comes to protein interactions, you have to do a lot of work before you complete your research study. So you might have to do a lot of analysis using other ExPASy tools, and if you combine them all together, you should be able to conduct a thorough research on the topic that you're working on. Okay, and once you use this, you should definitely cite this resource publication. So they are also giving the reference to the paper right over here. So if you click on this particular tab, you can see the publication, and then you should cite other publications, because that is very important for the authors to give them the credit they deserve, right? Thank you so much for watching the video. I'll meet you in the next one.